Good afternoon, everyone. My name is Rose Makovsky, and I'm the Director of Equity, Diversity, and Inclusion at Montgomery County Community College. It is my great pleasure to welcome all of you, faculty, students, staff, board members, citizens of the community, and beyond, to our 12th annual Presidential Symposium on Diversity. We created this capstone event in 2010 to provide an opportunity for ongoing dialogue on diversity and inclusiveness among students, faculty, staff, and the community at large. We've invited prominent and diverse leaders to speak to areas of relevant social, national, and academic interest over the last 12 years. In conjunction with this year's symposium, and because of COVID, we held a public service announcement contest in which all Monco students and employees were invited to create a public service announcement of their own in order to promote the importance of COVID-19 vaccines and of being vaccinated. At this time, it's my pleasure to congratulate student winners Ebony Carter, Peyton Myrie, Abigail Lichtfuss, William Moser, and James Duffy, whose winning entries were just displayed while you were waiting and will be on display, on prominent display in the days and weeks to come, but you saw them here first. Our college is a proud participant of the White House and U.S. Department of Education's COVID Challenge. As a designated vaccine champion college, we are working with colleges across the nation to encourage students, employees, and community members to get vaccinated. And as part of this vaccine initiative and as a continuation of our dialogue on diversity, equity, and inclusion, we are delighted to present to you our keynote speaker who will help us understand more and more about the science behind the vaccine, its creation, as well as its importance in protecting everyone. But before we do, one important housekeeping note. We are in webinar format, but your questions are important to us. If you will use the Q&A feature on your screens and type in your questions, they will be picked up by my colleague and yours, Ian Campbell, and posed to our speaker at the end of our presentation. To announce this year's symposium speaker and our very special guest, I would like to introduce our president, the person who puts president in Presidential Symposium of Montgomery County Community College, Dr. Vicki Bestecki-Perez. Vicki.
Thank you, Rose, and thank you for your leadership in the Presidential Symposium on Diversity for the past 12 years. I welcome everyone to today's program. And it's my pleasure to have the opportunity to introduce to you our keynote speaker, Dr. Kizzmekia Corbett, the lead scientist for the team that developed the Moderna COVID-19 vaccine. Dr. Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases at the U.S. National Institutes of Health, describes Dr. Corbett as a scientist who is widely recognized in the immunology community as a rising star and whose work is having a substantive impact on ending the worst respiratory disease pandemic in more than 100 years. Dr. Corbett's interest in science started at a young age. While she was growing up in Hillsborough, North Carolina, Dr. Corbett was fortunate to have teachers who recognized her curiosity for learning. Through her encouragement, along with her mother's guidance, Dr. Corbett participated in a program called Project Seed, spending her high school summers as an intern at research laboratories. This STEM experience fueled her interest in science, leading her to the University of Maryland on full scholarship to study biological sciences. Then she continued her education at the University of North Carolina, earning a PhD in microbiology and immunology. As the scientific lead for the coronavirus vaccines and immunopathogenesis team at the National Institutes of Health, Dr. Corbett spent more than 6 years with the team working on vaccines for coronaviruses, building on the decades of work to understand how the viruses function. Using the valuable knowledge, the team was able to develop a vaccine using the sequence for COVID-19. Currently, Dr. Corbett is an assistant professor of immunology and infectious disease at the Harvard Radcliffe Institute and Harvard T.H. Chan School of Public Health. She also devotes much of her spare time to advocating for vaccines and STEM education, often in communities of color. It is truly my honor and privilege to have this world-renowned viral immunologist as the keynote speaker of the 12th Annual Presidential Symposium on Diversity at Montgomery County Community College. Dr. Corbett, thank you for being our guest and sharing your experiences with us.
Thank you, thank you, thank you. And I must admit that I am sitting in front of my new computer and I've tried desperately to turn off my notifications, but they keep binging. And so I apologize in advance for that. And I thank you all for inviting me to give this symposium this year. It is an honor by far. And I will say that you all were one of the first universities and colleges that reaches out to me before the hype. And so I am thrilled to be giving the seminar. And I am very happy to also see that the students are taking such an active role in trying to campaign for people to get the vaccine, because that is where we are now. We are at the point where we do have these vaccines. They work beautifully and they're effective and they're safe, but really getting the message out and the education to the community is something that I feel like is beyond even me and is going to take a village. So to all the students who participated in the campaign, thank you for that. I am here to give background information on what is the race to a COVID-19 vaccine, to talk about the fundamental and foundational science of the coronavirus vaccine, particularly the one that I was leading a team to develop at the National Institutes of Health at the Vaccine Research Center, but then also to give a backdrop of all of the things that are happening around the COVID vaccine now and some of the data with that. And so I am currently an assistant professor at Harvard in the T.H. Chan School of Public Health. And so I bring you greetings from my new office there. And I wanted to start because one of the things, especially when I talk to universities, and particularly in crowds such as this, where is heavy on the student side, I like to start by talking about my not so rapid development of a scientist, because I think as bursting onto the scene, there's this idea that science happens very quickly and becoming a scientist happens very quickly, and that's just not the case. And I'll give the information around the vaccine, but for me, the trajectory to becoming a scientist was something that started a very long time ago, back even when I was in high school, and took a very windy road through the point of where I went to undergrad, and it was at the University of Maryland, Baltimore County, and then up to getting a PhD. I even took a year off between getting a PhD. And then I did a fellowship at the National Institutes of Health. And on the scale of how long these, what are called postdoctoral fellowships, how long they usually are, I took the longer route and actually did 6 years at the National Institutes of Health before getting to the point of where I am now at Harvard. And so that comes to serve as a bit of inspiration for any of you in the audience who are interested in STEM, that it is a long road, but of course, at the end, it is certainly worth it. At the National Institutes of Health, where I was then when we started to do the coronavirus research, the portfolio of viruses that are studied there really go from A to Z, or AIDS to Zika. And HIV and AIDS are actually the foremost, it is the foremost virus that is studied there. And then there's this portfolio that's very broad. And you can see right directly in the middle of all of these viruses, including some that are emerging pathogens, such as Zika, there is the SARS and MERS viruses. And I chose to work on these viruses largely because it sat in the middle and is understudied not just from the vaccine research center's perspective, but also at that time from the global perspective. And it was important to me because it was starting to spread in 2014 when I got there. And I chose it because it was high profile enough that I felt like I would be able to develop a really independent research career in it. And at the same time, be able to watch the trajectory of these very great vaccines that come out at the center go from the point of sometimes even a protein on the screen, like you see here, all the way through to advanced development. And now I'm here at the School of Public Health, where I study viral immunology for MERS coronavirus and other coronaviruses. I understand the pandemic potential, particularly of other viruses within a nidovirus order, and explore the prototype pathogen preparedness therein. And I came to the School of Public Health because one of the things at this moment really exemplified for me is that not only do we need to do the science, but we also need to very much prepare for vaccine equity and access, and also vaccine education. And then there's a multitude of other things, because I know that all of those things sound boring to people who are, newer and younger and more cool than me. But there are so many other opportunities that scientists have that a lot of times people don't talk about. But one thing that came in this moment is realizing that there are scientists on TV just about every single night, offers to speak at places like this and beyond, and et cetera. And so let's talk about the science and the rapid development of the COVID-19 vaccine. So firstly, coronaviruses are a large family of viruses, just like your family. There are cousins, there are brothers, there are sisters, and there are a lot of viruses within the beta coronavirus family, 7 of them to be exact, that have infected humans. We know mostly about the MERS and SARS viruses that have caused small epidemics, and particularly for MERS that caused over 2,000 cases worldwide. And MERS actually erupted around 2013 when I started to go into my postdoc fellowship. So we knew that this could happen. We knew that they had this pandemic potential, and the type of virus that could particularly be poised for a pandemic actually was noted to be a SARS-like virus. And that type of viral evolutionary biology was done by Ralph Barrick's laboratory. So I like to think of the 2019 emergence of SARS-CoV-2 into a global pandemic as basically the virus's way of saying, I told you so. We are about 5 million deaths. I'm sure we'll get there in a couple of days around the globe for this particular virus. And it really is a matter of us really not necessarily all the way paying attention to the signs that were in the history of this viral family. So where we are now is that this really ends with most of all of us being immune. And I take that verbatim from a quote that was spoken during an interview that I did. And you know what, I'm not even sure that I turned on my sound, but there was an interview that I did for PBS that really exemplified the idea that in order for a global pandemic like this to be squashed, that we really have to be immune to some extent. Now, if we count on the virus to make us all immune in this entire world, over 40 million people would have to succumb globally. And we certainly do not want to have to see 40 million people die before we get to the point where we are immune enough that this virus is less of a pandemic and more kind of matriculating like an endemic coronavirus. And so we prepared for this moment so that we would have a vaccine to develop in the case that a coronavirus emerged into a pandemic. And the way that we did this is under this umbrella of research called pandemic preparedness. And pandemic preparedness really comes with the idea that you think about a virus within a given family, and you do enough research with that virus in that particular family so that you can have a fast and reliable and universal vaccine solution. Now, what does that mean? A fast vaccine technology is something that can be produced in vast quantities quickly, Reliable vaccine technology is something that upholds a reliable manufacturability standard. And then universal is italicized because this is a vaccine technology that must be preemptive and provide future protection against future outbreaks. And while we are not there yet, we are certainly to the point of which we got a universal idea that we could really transplant onto the current SARS-CoV-2 virus. And so the way that we did this is by going about it with what's called the prototype pathogen approach. So prototype pathogen, that means a known pathogen within that viral family. We use MERS, which was, again, starting to circulate around the time that I started my research fellowship at the Vaccine Research Center. And you do enough research on that known prototype virus within that family so that you generate enough knowledge that it's applied to other viruses within that family. And ideally, you would want to develop your countermeasure, namely here, a vaccine, through a phase 1 clinical trial so that you speed up the development process shall a pandemic occur. This entire prototype pathogen approach to pandemic preparedness, I like to sum up, as I did in the New York Times, as the plug and play approach. And so how do we get here? This is the rapid development timeline for the SARS-CoV-2 mRNA vaccine, particularly the Moderna vaccine, where in the onset of the pandemic, towards the latter parts of 2019, we all heard about this respiratory virus outbreak that was in China, and people were getting sick. Some people were dying. It became clear that it was circulating from man to man. So we had a problem. And the sequences of that virus became known on January 10th of 2020. It was then called the 2019 N or novel coronavirus. And then only 66 days later did we go into a phase 1 clinical trial. All of that is summed up in a very expeditious way, right? Everyone's like, there's no way they could do this, but there's a large amount of work, even beyond the years stated here, but particularly between the years of 2013 and 2019 that went into understanding MERS coronavirus and how to make a really good vaccine for it at the Vaccine Research Center. And so what we did is we honed in on this one protein that sits on the surface of coronaviruses. The reason why coronaviruses are called corona is Latin for crown, and these spike proteins that sit on the surface really form a crown around that virus. And the entire purpose of that protein or main function of that protein is to really bind to the host cell, as you can see here. And then once it binds to the host cell, it allows for the virus to go inside the cell, take up that cell, and then replicate. And that's how you get an infection. And then subsequently, that's how you get COVID-19. And so if you want to build up an immune response to the virus, the idea is that you don't necessarily need the whole virus. You really just have to teach your body how to recognize that spike protein. And so we wanted to do that. But in order to do that, you have to know as much about the spike protein as possible. And so we went about advancing our just basic biological knowledge of the coronavirus spike protein. And in order to do that, you have to be able to see it in high definition. And so in collaboration with structural biologist Jason McClellan and cryo-EM expert Andrew Ward, we were able to publish the first high definition structure by cryo-electron microscopy of a coronavirus spike structure. And at the same time, another coronavirus spike structure was published, and then the literature kind of just lit up with multiple labs around the world defining the structure and the function of these coronavirus spike proteins. And there came a time where the spike proteins for MERS and SARS, which were the most epidemically relevant, those are the ones that most people really cared about, were defined by George Gao's group in China, and then also our group at the Vaccine Research Center with all of our collaborators. And we defined that structure with our collaborators really with the understanding of how you can make it into a really good insert for a vaccine, or as it said here, antigen. And so we did that because we knew how to rationally design this protein to make it into its best self, so that when you present it to the body, that is exactly what you want the body to learn from. And so it's almost like making the recipe for your body's immune system to learn from. And so we did that by looking at the regions of the spike protein that are important for it to do its dirty work. So the entire purpose of this protein, as I said, is to bind to the host cell and then allow for the virus to get into that host cell. And it does this through a process called fusion. Fusion happens when this little spring, as you can think about it like the Slinky toy that you may have played with as a kid, or maybe I'm dating myself, I'm not sure, but like a Slinky toy. And when this spring pops up atop of itself, that's when fusion occurs. And when after fusion occurs, that protein actually isn't even in its best self anymore, particularly as is noticed by the immune system. So you want to lock this spring down and present that protein as a pre-fusion or before fusion version of itself. And in order to do that, us and our collaborators in Jason McClellan's laboratory and Andrew Ward's laboratory, we scan this protein for regions where we might be able to add mutations to the protein to make it, again, a better vaccine antigen or insert. And we came across 2 proline mutations that sit here in the apex of the fusion, the central helix and the heptad repeat 1 region. And that protein called the MERS S2P or 2 proline was effectively stabilized in its pre-fusion conformation, which is kind of this mushroom-shaped version of the protein. As opposed to when you express it without those 2 proline mutations, it's more flimsy. And again, it flips upon itself and causing the post-fusion conformation, which is a more elongated. So this is the protein that you want to give to your body to make the best immune response that you can. And so we did all the work to understand that immune response, and a lot of years went by, and then we came and started to collaborate with Moderna, which is a company that uses messenger RNA to deliver various therapeutic proteins. But in the case here, because we wanted to go fast, and because we wanted a vaccine technology that was reliable from a manufacturability standard and universal enough so that, as I said, we can plug and play our particular protein into it, we thought that this would be a great platform in case there was a pandemic. And so we inserted that MERS S2P protein into their platform, which basically is purified messenger RNA that is encapsulated into a lipid or a ball of fat. And then when that is delivered into a person, your cells read that message, make the spike protein, and then your immune system learns exactly what that spike protein looks like without you having to be infected. And so the technology is fairly revolutionary and also very, very brilliant. And because the reason why we were able to go really fast is because we had used the technology in the mouse model to serve as a proof of concept for this type of delivery. So we inserted that MERS S2P protein into the messenger RNA, and we delivered it to mice that, unfortunately, when they are not vaccinated, they succumb or die from their MERS infection after 8 days, as you can see in the gray group. When they are vaccinated, these mice live and do not have any much weight loss, particularly in a dose-bearing way. So even down to a 0.01 microgram dose, which is so small, you can protect these mice from succumbing to the virus. And also, that was not just seen with weight loss, but with the amount of virus that was in the lungs of these mice and the amount of lung hemorrhage, which is a score for the amount of lung disease. So this was a proof of concept that delivering of the spike protein by messenger RNA at least worked in a preclinical model. And focusing on that green bar, that is a very low dose, it actually still, while it may not have protected that well, it didn't cause any enhanced illness, which is something that a lot of people are particularly concerned about from vaccines. And so this was a very good sign. And so we got to that point where we could conclude that we could deliver these very immunogenic, stabilized spike proteins via messenger RNA, and that they elicited really, really good immune responses or antibody responses in preclinical models or animal models. And we could transplant that into the SARS-CoV-2 and deliver that by messenger RNA as well. And because of all of that work, it really led to the unprecedented speed of what we call the mRNA 1273 development, and also informed the global vaccine portfolio. And I know it seems like I'm skipping over so much science, and that is actually the case because so much science went into the preclinical steps of this particular of this vaccine development in order for it to even be authorized to go into a phase 1 clinical trial. And I don't necessarily talk about all of that work, but I really want to just highlight very briefly that we study how this particular protein is expressed in a dish of just cells. We vaccinate mice, we vaccinate monkeys. We really look in a very detailed way at the types of immune responses that happen in these animal models prior to going into a phase 1 clinical trial. And at the same time that we were doing that, Moderna had initiated what is called good manufacturing practice of production of the clinical grade material to go into a phase 1 clinical trial. And they were able to do that in only 4 days following the sequence release of the virus. And that allowed for them to ship it and ship the clinical material in only 41 days following that, which goes back to this type of platform being fast and reliable. And then the phase 1 clinical trial, which happened in 66 days, started soon after. So let's skip a little bit ahead, just again, because of limitations of time, to the point of where we get to where we have phase 3 efficacy results. I just want to remind everyone, even though it seems like forever ago, that these phase 3 clinical trials, not just for the Moderna mRNA 1273 vaccine, but for all the vaccines, are controlled in a way that people get half of the people in the vaccine trial get a placebo, in this case, 15,000 people, and then another 15,000 people get the vaccine. And then over time, they're followed for the amount of COVID-19 incidents. And that is how you come to the point where you could say this vaccine is effective at 94.1%. And because there were only 11 cases of COVID-19 in the people who got mRNA 1273 compared to the 185 that got the placebo. There are, of course, and one of the things that is important as you guys are campaigning around people getting the vaccine is to be very upfront about what are called adverse events. These are in vaccinology what we call a reactogenicity. So this is your body's reaction to getting a vaccine, and it's not actually a bad thing for you to get maybe swelling or pain at the injection site or fever or headache, because it's your body-based immune system saying, okay, we're awoke now and we're ready to fight for you. So in conclusion, following those promising phase 3 efficacy results, mRNA 1273 is currently being used in multiple countries, obviously including here, and is a part of this global vaccine portfolio where over 6 billion vaccines have been given to date. And one would think that we would just be able to drop our mic and walk away. But what about now? The science continues to happen. We continue to answer very critical questions about the science of the Moderna vaccine, but then also about many other vaccines as well. Just to name a few of the questions that we're continuing to answer. So it's firstly, the immune correlates of protection. So this is our way of saying what type of immune response correlates for protection to allow us to inform how to make the really good vaccines in the future, obviously, but then also to inform other vaccines that are in the global portfolio to reach kind of a mark of the amount of types of immune responses that they might want to hit in order to be distributed around the world. And then there is this idea that time has passed, and so what about the durability of protection? And so on an ongoing basis, people from those phase 1s and phase 2 and phase 3 even clinical trials are followed so that on an ongoing basis, the durability of the protection from those vaccines, particularly the mRNA 1273 vaccine, is published. And most recently, durability was published in Science this year. And then there is the variants, which everyone is concerned about right now. I think the hot topic is the Delta variant, and then some people say it's the A4 blah, blah variant in the UK. Nevertheless, we are in constant engagement with trying to understand how the vaccine protects against the various variants that pop up. The most recent of those publications was published in Nature Immunology, and then boosting. And I have that in red because I'm going to tell a story of how we come to the point of where we understand what types of immune response follow someone getting boost and why the recommendation in certain populations to start to boost at this point has been given. So this particular graph shows the immune responses in people from the phase 1 clinical trial. And this time point is a snapshot that is highlighted here, is a snapshot at that 2 weeks after someone gets 2 doses of mRNA 1273. So when you become fully vaccinated 2 weeks after your second dose, you have a very high antibody response that is called a neutralizing antibody response that can effectively, kill or prevent a virus from getting inside of the cell. And because we wanted to see what the types of immune responses were also in older people, we also immunized older people above the ages of 56 and showed that those immune responses after 2 doses were on par with the people that between the ages of 18 to 55. So mRNA 1273 elicits a very potent neutralizing antibody response, and that's great because antibodies are part of the immune system and package that you need to protect yourself from COVID-19. Now, as time has passed, as I've said, if you follow that blue line, this is the 18 to 55-year-olds. And so there is some waning of the immune response, but still the antibody responses are fairly high. But there is a more steep slope when you look at people above the age of 56. And so in order to prepare for the moment, and that's not just for the ancestral strains of the virus, or so some of the earlier strains, but then also for some of the strains that are circulating now, like in particular in the magenta, the Delta strain. And so in order for us to say, do we need to boost those people? And from where that recommendation comes, many people are right now out to about 6 or more months following their second dose, particularly those people in the age population, because they were among some of the first to get vaccinated. And so it has been recommended that older adults and other at-risk individuals receive a booster. How do we get there? How do we get to that point of where we can use science to make those types of recommendations? I'm going to show you just a snippet of the story where we were understanding the utility of boosting in the non-human primate model. So rhesus macaques are a non-human primate, and you can immunize them and follow them after being challenged with a virus to look at the amount of viral replication, both in their lungs and then also in their nasal cavity. And so we immunize these monkeys in the same way that a person might get immunized if they went to their local CVS, with 2 doses of the Moderna vaccine, week 0 and week 4. 6 months after that, and kind of mirroring what's happening in people just about right now, we came in and boosted those animals with either the ancestral mRNA 1273 or an mRNA 1273 that has a spike protein insert from a variant called the beta variant. Again, to remind people that while the Delta variant is currently the one that's most circulating, the beta variant is the one that evades or hides from the vaccine immune response the most. And so it's really was setting a bar high for this type of experiment. We also have this group in the light blue where these monkeys got 2 doses of just the mRNA 1273 with the beta variant insert. We came behind in those animals and we challenged those animals with a high dose of a beta variant to ask the question, was it better to boost with the ancestral mRNA that's going to be in the red on all of the slides, or variant mRNA, which is going to be in the dark blue on all of the slides? And what we saw, particularly in that light blue group, which was kind of the control group, is that the immune responses from the mRNA in that control group were really on par with what we've seen previously in our animal studies for just the ancestral mRNA. So what you're looking at here is the timed response of the antibodies that bind to the spike protein and also to the receptor binding domain portion of the spike protein, as they are to the ancestral strain, and then also to, in the red, to the beta strain. So after 1 dose, you get a increase in antibodies in these monkeys. And then of course, it's boosted by about half log to a log after the second dose of the vaccine, which is exactly what you would expect. And at that point, about week 37, when it's time to challenge those animals, the animals, just like in the humans, have about 10 to the 3 neutralizing antibody responses. So they have high level antibody responses in that control group. But what about looking at the groups where we let the immune response wane for 6 months following their 2 doses? In this particular group, they got mRNA 1273 at week 0 and then week 4. And then at week 6, you can see the peak immune response. Particularly if you look at the Black, this is one of the ancestral strains. And so that immune response in these monkeys at peak is about 10 to the 3, 10 to the 4. Over time, just like as you would expect, there is some waning of the neutralizing antibody response. And when you boost with the mRNA 1273, you see that the immune response or the antibody response is reignited. And it's not just for the ancestral strain, which is in Black, but to many of the other variants that have circulated around the globe, including in the dark blue, the Delta variant. And so that was one of the first clues that when you can boost, you can just re-excite the immune response and take it back to actually above the point, as you can see, above the point of where it was at the previous peak. So it's almost like we wanted to reset the immune response in these animals. And that happens not just with giving the ancestral mRNA 1273, but also if you give mRNA 1273 with a beta insert. And so boosting increases the antibody responses both to the homotypic, so the ancestral strain, and then also to variant strain. So that was really good to see from the antibody response perspective. But then we wanted to look at B cells. So B cells are the cells in the body that make antibodies. And so looking at the type of specificity that you have in your B cells, and also how fast they might be able, you can really recall that specificity following a boost would be important to see. So we looked at 3 different types of B cells when we sorted them out. The first of which are B cells that are just specific for the ancestral strain. That is in the white. And the gray are the B cells that are specific for both the ancestral and also the variant strain. That is in the gray. And then in the Black are the B cells that are specific for the beta only, or the variant strain only. So zoning in here at the bottom into this group that is the control group that only got the mRNA 1273 with the beta insert. You see that a large proportion of this pie slice is Black, as you might expect, because they got 2 doses of a vaccine that has the beta insert. So their B cells are in a great proportion beta specific B cells. Interestingly, after 2 doses of mRNA 1273, what you see is that a large proportion of the the B cells are both and have specificity to beta and also to the ancestral strain. So they are double positive, and that means that they have broad responses. And those responses don't necessarily really wane over time. So those B cells are kind of just hanging out and can respond to either beta or Washington, or specific for either beta or Washington. And then following a boost, you can see that there's no expansion of any one type of B cell or another. So the double positive B cells are still remain. So what this says is that boosting either the homotypic or the heterotypic mRNA 1273 elicits memory B cell responses that are double positive, and that boosting doesn't expand to any single type of B cell or another. And so you really are just maintaining these really broad B cell responses with the course of this vaccination. The same kind of happens with the T cell responses, although you can see that with T cell responses, the T cell responses, as we always have known, are more skewed in the CD4 direction, of course, and then also in the TH1 direction, which is the type of T cell response that you would want, because it is the type of T cell response that has not been correlated with enhanced disease before. And so when you look at particularly, let's start with the red group. When you look at the red group, which is the group that got 3 doses of just the mRNA 1273, at week 6, you see that there's a TH1 skewed response that does wane over time by week 29. But then when you boost, you can reboost that response just as you did with the B cell responses. It is notable also that the same is true for TFH cells, which are the types of T cells that help to get really good antibody responses. And so boosting re-stimulates the mRNA 1273 specific T cell responses as well. And so we looked at protection. One thing that must come of note here is that in these types of experiment at the time, this experiment was used to inform the policy of if we're going to boost, should we boost with the ancestral mRNA or should we boost with the mRNA that represents a variant? And so we don't have an unboosted group of animals, but what we do have, of course, as a negative control are animals that have not gotten any vaccine at all. And what you can see is that after they've been challenged, immediately after their challenge at day 2, those animals that did not get vaccinated have a large amount, up to 10 to the 6, 10 to the 7, viral replication in their lungs. Whereas the animals that have gotten 2 doses of mRNA 1273 with the beta insert as a control have completely wiped out their viral replication. So the vaccine protects, woohoo, we kind of knew that. But what happens in those boosted animals? In those boosted animals that got mRNA 1273, and then 6 months later got boosted with either mRNA 1273 in the red or mRNA 1273 with the beta, you can see that you basically wipe out in a majority of those animals any of the viral replication in the lung. And the response is down about 5 logs, actually, to be exact. And then with all of those animals, is not just that initial response is lower, but that the clearing of that virus from the lung, viral replication from the lung, is quicker. So at day 7, where there's still some virus hanging out in the lung of those control animals, there's really no virus in the lungs of the animals that have gotten the vaccine with the boost. And the same is true for the nasal swab. There is a little bit more variability with the nasal swab. And I'll talk a little bit about that. But nevertheless, with the vaccine and with the boost, you have about a 3 to 4 log decrease in the amount of viral replication in the nasal swab in the upper airway. And then, of course, by day 7, you've basically wiped out in a majority of the animals any of the viral replication. And so we look at that viral replication from 2 different directions. The first is what you just saw, which is a very highly sensitive method. This is a PCR method that looks at SG or subgenomic RNA, but then we also take the fluid from the lung and then also take the nasal swab, and then we try to grow virus out of that fluid or nasal swab to beg the question, does virus actually grow? So sure, you might have some viral DNA hanging out there, and it's very sensitive. But does viral RNA? But does the virus actually grow? And what you can see is that there's really no viral load in the lungs of any of those animals that have been vaccinated at all. Whereas there is about a 10 to the 6 amount of TCID50 in per milliliter of the fluid from the lungs in the animals that have not been vaccinated. In the nose, the story is a little bit different. Nevertheless, almost wiping out most of the viral load in the nasal swab, but you can see that there are some animals that do still have a little bit of virus that is in the nose. So when people talk about, oh my gosh, I've been vaccinated and then I got my nose swabbed and I had some virus there, you can see that it is basically breakthrough. And so having some virus in the nose is not something that we didn't know might happen. And actually how you think about it is that there might be this differential ability to protect in the lower airway, which is very severe disease, versus in the upper airway. While you do knock down replication, you might not necessarily be able to get what's called sterilizing immunity. And so overall, mRNA 1273 boosting in these non-human primates is highly protective against this particular beta variant. But what about breakthrough in the nose? So we actually saw that a lot in some of our initial studies. And because of that, we went about trying to understand exactly how we might be able to correlate protection in the lower airway and the upper airway by specifically designing an experiment that assesses immune correlates of protection. And so this particular experiment in the non-human primates was one that was particularly designed with various doses of mRNA 1273 given at week 0 and week 4. And then with a challenge modestly only 4 weeks after to ask the question, at which doses, which types of immune responses is protective versus those that are not. And what we knew or assumed via hypothesis at that time is that it would be the antibodies that bind to the spike protein that would correlate very well with protection. And so what did we see? We looked at both what are called humoral or serum antibody responses and also mucosal antibody responses. So those are the antibody responses in the lower airway, in the bowel, and also in the nasal washes. And what we saw is that as you took the doses of mRNA-1273 down, you got lower antibody responses. And these antibody responses that we assessed were a very wide array of assessments. We looked at antibodies that just bind to the spike protein. And then we looked in 3 different ways at antibodies that can neutralize that virus, neutralize the virus, at least in vitro. And the story remained the same. The higher the dose of the vaccine, the more antibodies you have. And so there's this dose dependency to the amount of antibody responses. And it was that we looked at the amount of protection that came from that and showed that the same type of dose dependency was shown in the protection. Whereas when you give 30 micrograms of mRNA-1273, you have 4 logs less virus in the lungs of those animals as compared to the control group. And then there's this stepwise amount of virus in the lungs as you decrease the amount of mRNA-1273. Of course, as the story has always been told, the vaccinated animals clear the virus from their lungs very rapidly. But what about the nose? You can see that pink group. The 30 microgram group is really the only group that had any notable protection or decrease in viral replication in the lung. When you got down to the 10 microgram group, those animals had the same type of viral replication or amount of viral replication as did the control animals. And so this became our first clue that there might be this disparate ability for the vaccine to protect in the lower airway versus in the upper airway. While it's still good, mind you, right? This is 1/3 of the dose that a human gets. And this experiment was designed to particularly test that, but it did become our first clue. And so then we looked not just at the antibody responses prior to challenging the animal with virus, but then also after the animals have been challenged, because what you can see is that in the PBS group, which really they didn't have any antibodies before the challenge, after the challenge at about day 14, and particularly at day 28, those animals had antibodies to the spike protein. And the only place that those antibody responses could come from is from the virus that they saw on during their challenge. So what about the animals that had high antibody responses prior to getting challenged? You can see that they really didn't have an increase in their antibody responses, which means that there is no real anamnestic response once you've blown out that antibody response prior to getting a challenge with the virus. This means also, in a roundabout way, that those animals that were protected, so to speak, with the high dose of vaccine didn't really see the virus antigen in the same way that the animals did who had very modest antibody responses and lower doses of the vaccine prior to getting challenged. So you can see that, like, for example, in the animals that only got 3 micrograms of the mRNA-1273, they actually had a very increase in the amount of nucleocapsid, which is another protein on the surface of the virus, responses following being challenged. And so this is a clue that there is this antibody dependent ability for the vaccine to protect the animals from seeing the virus antigen. And so it looked like at that point that there was about a 500 international units per milliliter associated with no increase in the N-specific antibody responses post-challenge. So we're starting to hone in not just on the fact that there was this antibody dependence of the amount of protection, but also at the just stark amount of antibody that one might need in order to be protected against seeing the virus antigen post-challenge. And so we did the standard correlations where we looked at the amount of spike specific antibody on the x-axis and the amount of viral replication both in the lung and also in the nasal swab, and show that there was a very strict inverse correlation. So the more antibody that an animal had, the less viral replication they had at day 2 following being challenged, which is something that was very clear from the previous slide. But moreover than that, we could show that for about 1 log in the amount of spike-specific antibodies, there correlates with about 1 log in the change of the viral replication in both the bowel and then also in the nasal swab. And that there was no animal above a certain threshold that had any real detectable viral replication. And so what that showed is that there was about a 1 to 100 or 300 or 600 for the nasal swab, a threshold to be able to show that, okay, these animals were protected versus those animals that were not. And so we were starting to hone in on the amount of antibody needed, but it became very clear to us that in order to say, we could of course say that antibodies are a correlate of protection, but are antibodies that are elicited specifically by the vaccine sufficient for protection, taking away all the other components of the immune system? And so what we did was we took antibodies, isolated those antibodies from those non-human primates that had gotten vaccinated. We purified those antibodies and then delivered them back into another model, the hamster model, that is a pathogenic model for SARS-CoV-2. And we did that at various doses of the antibody to show if there was to show if there would be a differential in the amount of protection in these particular hamsters, comparing it to, as you can see in the gray, to antibodies that were isolated but prior to the non-human primates being immune. And so what you see is that the hamsters that got a low dose or that got a control antibody were not protected against SARS-CoV-2, whereas the hamsters that got 10 mg of the antibodies that were isolated from the vaccinated non-human primates were completely protected against weight loss. And so this was a stamp to the fact that not only were antibodies a correlate of protection, but they were certainly mechanistically sufficient to protect against SARS-CoV-2. And so this slide I like to use to summarize all the things that I've said over the last, I think, 45 minutes, where we show on a sliding scale the amount of protection elicited by mRNA 1273 vaccine in the non-human primate. So when you start at the very high end, there's protection in the upper and the lower airway. This is because the antibody responses are very high, and that is where we were then, where there were very high antibody responses and protection in the lower and upper airway with the original SARS-CoV-2 strains. But where we are now is where some of the antibody responses have waned. So we're right about here in midway on the sliding scale, and some variants are circulating. And the vaccines are largely still protective against severe disease. And so for the most part, upwards of above 90, particularly in the 18 to 55-year-old age group, people are not presenting with very severe disease because there's still tons of antibodies available to protect you in your lower airway. Now, you might see some breakthrough infections, and those types of breakthrough infections and the differences in protection in the lower airway in the older and immunocompromised are why the boosters are being recommended. And so in closing, I would like to thank all of these people on this slide who worked with me over the last 6 years at the Vaccine Research Center. I would like to thank everybody on this slide who, actually, I don't even think this slide is complete, and I probably should update it, but everyone on this slide in some shape or form was critical in making sure that the COVID-19 vaccine response from the VRC went very smoothly and rapidly. And particularly Dr. Barney Graham, who served as my immediate mentor, and Dr. Cedar, who served as a mentor on the non-human primate studies. And all of the contributors and collaborators, firstly to Moderna for allowing us to work with them over all of these years, really in an academic direction to answer some really critical viral immunology and vaccinology questions. Jason McClellan's laboratory at UT Austin, Andrew Ward's laboratory at Scripps, who were instrumental in the initial structural design and structure studies for the spike protein and the design of the stabilized prefusion spike protein. University of North Carolina, Ralph Barrett's laboratory, who's done all of our mouse challenge studies to date. And then all of these other collaborators who have come on board, whether it be at Emory or Duke University, to help us with various different assays during this coronavirus vaccine development. And then, of course, A special thank you to all of the clinical trial volunteers for participating in the studies and being a data point on some of these later slides. And without further ado, I will answer questions.
Thank you so much, Dr. Corbett. You made me I've always respected science, but you made me have a completely newfound respect for science and scientists, and your passion for what you do is very, very clear. I wish we could do a round of applause or a standing round, but that gets kind of clunky in this format. But believe me, that was so informative and so helpful, and it puts a lie to the simplistic ways in which the vaccine is treated in the public. Thank you, really, thank you. We have some questions, and I'm going to pass it over to Ian Campbell to pose them to you. And if that's okay with you, we can get started. Ian?
Yeah, sure.
Hi, Dr. Corbett. Thank you very much. And thank you, Dr. Kizzmekia and Dr. Makovsky for bringing Dr. Corbett to us. What a treat. What I'm going to do, Dr. Corbett, is I'm going to read the question and answers that have been posted. And I know you can read them. However, the audience, of course, can't see them. So I'm going to read them out loud to you like we're doing a question and answer. And that's how we'll do this. My first question comes from Bernard. He says, despite the wealth of information currently available, many remain resistant to taking the vaccine. What's your elevator speech for encouraging those holdout family members and friends who remain unconvinced about taking the vaccine?
So I don't start with an elevator speech. I always ask people what their specific concerns are and then address that very specifically. Because I think that at this point, about close to 2 years since the vaccine development started, people who are still hesitant, or as I like to call it, inquisitive about the vaccine, have a particular issue, or they've heard a particular thing. And so I just like to empathize with whatever it is that is very specific to them. And generally, when I'm giving kind of an elevator speech to people who I don't know whether they've been vaccinated or whether they are inquisitive or not, I start with saying, this vaccine, while the technology seems new to you, is not new to us on the science side. And it was started in development several years ago. And that is because we understood so much. The reason why I went so quickly is because we understood so much from our MERS coronavirus efforts. And did you know that some of that data was published even prior to this pandemic? And that kind of changes the perception of that the vaccine was developed really quickly. And then I say, what you're seeing now is not just an amass of all of the knowledge that came before the pandemic, but really some cooperation between, government and academia and industry in a very global way to make sure that this vaccine process went very smoothly and quickly, but without any sacrifice of the science.
One of the ones I think we hear about in the media a lot, this is a follow-up question, is the blood clot concern. Because that, I just saw the face you made. But can you speak to a bit of someone who only, as to maybe people who only get the media version of that? Can you speak to what that even is and what that's about?
So there was some blood clot concerns, particularly from the J&J vaccine. And what I can say about most things that are a concern is that the way that the FDA evaluates whether or not the vaccine is still safe for mass use is by looking at the numbers and looking at that particular condition and the numbers of that particular condition in, say, unvaccinated population versus the vaccinated population. And so when you compare those numbers, it becomes very clear that regardless of very, very, very extremely 0.001 or something percentage of people who might get a blood clot after getting a vaccine, that number is higher if you get COVID-19 and are unvaccinated. And so whenever you are weighing basically any one person who's deciding to get a vaccine is making their own personal risk assessment. I try to frame it around this in the same way that we frame it from a population standpoint. what your chances are if and when you come in contact with COVID-19 are always going to outweigh that of if you got vaccinated.
Oh, I mean, right, of course. What a great way to put that. My next question comes from Stephanie, and we're going to go through the Q&A first, and then we'll come to the chat after that. But Stephanie asked the question, she's curious to know how scientists determine dosage. Now that we're working for vaccinating our youth and maybe ages 2 through 5 and even from birth, if that'll be recommended. Is this something calculated or determined through trials, just like we saw? Can you talk a little bit about that?
Yes, actually. So the 100 micrograms that I continue to say over and over and over, and that goes into your body and also went into those monkeys, was determined via trial. So the phase 1 clinical trial, there are some main objectives of the phase 1 clinical trials, which is generally, it's first in humans, so it's about 50 people, and you ask the question, what dose, how safe, and what types of immune responses? Very simple question. It really is a very small safety study. And so you look at different doses. And in the phase 1 clinical trial, it was 25, 50, and 100. And 100 micrograms was determined to be the best dose for people above the age of 18 because the safety profile was very good. I'm sorry, it was 25, 100, and 250. I'm sorry. And 100 was determined because the safety profile was really good. And you got consistent immune responses following the vaccine. And the same way with the children, right? The doses, as they take the age down, they also take the doses down in those trials to pinpoint the right dose for that age group. And then I say that in the context of humans, but we do it even more intricately in animals because animals are more plentiful. So,
right,
yeah.
Right. Thank you. Sam asks, Harvard and NIH are pretty rare, he says. How many other groups are able to do the advanced study for mRNA platforms and timely responses of specific vaccines? Are any in regions where people live in close contact with host animals or have economies more reliant on manual labor?
So that's a large question. And the truth of the matter is, I think it is very clear that we took a very fortunate stance. I cannot deny that one bit, right? There's, Moderna is like right down here. And, the NIH and funding and, tons of scientists who kind of already had the knowledge and the database and the tools, agencies working together, all of these things, right? That being said, there are some other places that did develop their own vaccines in, I would say, very good speed and in very clear, safe and effective vaccines. So like Taiwan, for example, has a vaccine. It's from a company called Metagen. It's a protein-based vaccine, and it didn't come on board, as quickly as the messenger RNA, but they used the technology that they had to get to where they are now, which is, I forget the amount, but a large, overwhelming percentage of their population is vaccinated. And so, mRNA technology, one thing about mRNA technology is because it's so new, I fully expect that, but not that it will start to erupt on the scene in various different markets. But one of the things about vaccine access is, of course, going to have to be able to get technologies to various corners of the world. And I think yeah, anyway, and the part about the animals, I ignored that because it was extra.
I think one thing that a lot of us have been hearing is that there's boosters coming online for people, and then there's so many parts of the world that haven't received any vaccine. What do you think the key is to getting the vaccine to those corners of the world?
So I always said that my job was easy, even though it was not. But on the scale of jobs that people had throughout this pandemic, there was, developing the vaccine, regulation, which the FDA is in charge of. Could you imagine being like in charge of like telling people that this vaccine is safe and effective, and then distribution, and then a multitude of other jobs in between. Oh, needless to say, I think that some of those other jobs are far harder than mine. And so I don't like to tell them how to do it. There are people have said various things, right? I know the things that are happening, albeit maybe not to some people's standard or as quickly as we would all like. But as a reminder, we are only 2 years inside of a global pandemic, and we have a vaccine to even think about being critical about distributing, which is a blessing. And so, you know, increasing manufacturability across the globe, putting a little bit more burden on some of the wealthier nations to make sure that other people around the globe are vaccinated, being very strategic about where and how you vaccinate, right? Because there's so many billions to billions of people around the globe, and certainly not enough doses to go around. And so, you know, being very precise about how and where you vaccinate. And you're right. Part of the reason why our boosting plan takes a more modest slant than maybe some other countries is because that's true, right? There are populations that if they were to get COVID, albeit some waning of antibody responses and variant circulating, that would not get really sick. And maybe we should not be boosting them right now so that we can get vaccine doses out to some people who have not even seen one dose at all, which is a large proportion of the globe. And so, you know, equity of vaccine access sits on my spirit very heavily as a burden, whether it be inside of this country, which we've done a good enough about, and also globally. So it's going to have to be basically pulling a lot of muscle. And you know what I say all the time? I say, if Amazon can get you a package in 2 days, like, I actually just ordered a dress to be shipped to LA for like a gala, and it's going to come there Saturday, and like, yay, Amazon. But like, we have the types of systems in place. It really is just a matter of the planning and the preparation to make sure it happens.
Yes, yes. My next question comes from Stephen. He asks, were conclusions of your research shared with other pharmaceutical companies, and were theirs shared with Moderna in an effort to move quickly develop the vaccine? I think he's asking, how did the sharing happen in the development?
Openly, maybe too much so, but I'm, you know, very, very, very openly. I mean, you know, our research, we were very transparent about it very early on. And for example, the spike protein with the 2' mutation is being used by so many different companies around the globe, including Medigen, like I spoke about, in Taiwan. Pfizer uses it, you know, and so it was a very open, I wouldn't necessarily call it sharing, because that feels like we all got in a conference room and just like, yay. But no one was really saying, you can't use that, you know. It was very open. Right, right. And in a way that was atypical of how companies normally work towards this, correct? You know, I don't know what's typical. I've never experienced this before. That's fair. Yeah, that's fair. Right. I would say that anyone who is really invested in vaccinology and like the core of their spirit takes the stance of a free for all approach to this. Yeah, yeah, yeah. That's actually wonderful to hear. That's wonderful to hear. My next question is from Jenny. I think a lighthearted question, but I think she means it. Is there any research around the need to vaccinate domestic animals? And I think she's asking for the dog currently sitting on her lap. We will not be vaccinating your dog, but I called to vaccinate camels, for example, in Saudi Arabia with MERS, because there's no real human to human transmission, but like camels are very specifically transmitting MERS even today still to people. Huh. But no, there is no real necessity to vaccinate any animals at this point. Great, thank you. All right, Sasha has a question. How do you feel about boosting with different vaccines? Like, how does that, do you get a stronger immune response? She's curious about what happens. You get a great immune response. That data came out from the National Institutes of Health, I think maybe 2 days ago, I think. Yeah. And I actually tweeted about it last night, and I rarely have time to tweet anymore. So you can check out my thoughts on Twitter. But I mean, listen, boosting with a different vaccine is very safe. That's what the small study showed. And also you get very robust immune responses if you boost, for example, someone who got J&J with either of the mRNA vaccines, and you get a very strong immune response if you use someone with Moderna with Pfizer and Pfizer with Moderna. And so I think the data is very clear, and I would say that if it is your time to be boosted, don't be picky about your boost, just to do it.
That's great advice. Thank you. Stephanie asked a question, which I think you've talked about a bit already, but I love the way she asks it. So I'm going to ask it. Based on what you've seen in public discourse in the last couple of years, what is something that you realize that most people don't understand or know about the immune system and vaccines, but wish that they did understand?
I think that people don't know vaccine history. I think that we haven't gotten to the point. We didn't do good about telling people why they were vaccinating their kids. We didn't do good about telling people why they should get the flu vaccine every single year. And so we came into this moment and everyone's like, oh, but it might cause long-term effects. But like that never happens. So why would it happen here? Or people would say, you know, I don't want to get, it's not safe. Like vaccines are not safe for pregnant women, but actually they are very safe for pregnant women. And in fact, part of the reason why your doctor begs you to get a flu vaccine if you're pregnant is because you pass those antibody responses off to your children. And the same thing for these COVID-19 vaccines. And they're far more safe than getting a virus if you're pregnant. And just, those types of details about vaccinology in general are just, we didn't do a good job about teaching the public about vaccines. And so vaccine awareness and also decreasing vaccine so-called hesitancy is going to have to come with some continued conversation, not just about this vaccine, but all of them.
Yeah, Dr. Corbett, just a generation ago, we developed a polio vaccine. And in that time, we did a measles vaccine. What has been forgotten about what those vaccines have done to us? Is it a generational memory?
The disease has been forgotten. No one is getting polio, and most people are not getting the measles. And so someone said that early on in the pandemic, and I forget who it was, but they said, the good thing, the very odd thing about public health is that if it works well, no one recognizes that it works. And then if it doesn't work, everyone's criticizing it. And it's the same thing with like vaccines. It's like we forget why so many things are, so many diseases are largely eradicated due to vaccinology. And, we forget some of those very basic vaccines that we have that prevent us from getting childhood illnesses and have double tripled our lifespans over the last century. Vaccines save lives, and I don't think that's clear enough because we don't necessarily see what's happening. I think it also comes, that kind of thing is also why I was recently at Emory University, and I, just was walking around and I was like, oh yeah, how's your ICU doing? And they were like, oh, it's a mess. And it's like everyone outside of the hospital is just walking around their normal day, and inside, there's a pandemic happening and we don't see it. And so it's hard for people to grasp the concept of the necessity of vaccines because it's not necessarily in our faces. And I think that's, the problem.
Yeah, an amazing thing given the ubiquity of all types of media. I have a question from Patrick here. He asks, most of the data seems to use antibody blood levels as the marker for immune response. What about T cell and memory B cell responses? Do you look at them as a measure of vaccine response? And could the difference between them and the body's antibody complicate the understanding of how much immunity someone has from a particular vaccine, especially over time?
Definitely over time, yes. But I think antibody responses are very much sufficient and the correlate of protection. That's kind of what my last couple of slides showed. T cell responses, they are good because T cell responses probably work down in your lung and attack the virus and, you know, kill off the virus if you ever do get infected. And, you know, B cell responses are good because the recall of B cell responses make very rapid antibody responses if you do get infected. So having those like resting memory B cells that are specific to the antigen or the spike protein are very good. And we know all that. And we've detailed that several times over. Even a paper today coming out in Science, we talk about that. My paper today coming out in Science, we talk about that.
Right.
But the reason why antibody responses as a measure of the amount of protection is such a thing, number one, because of the easeability of the measurement. And then also because it's very clear that as antibodies wane, protection goes down. And so if you're going to assess the situation, assessing the basic antibody responses is the way to go.
Great, thank you. My next question comes from Mike. He asks, are the Sputnik 5 and the Sinovacs, which he calls them, deployed earlier in Russia and China, respectively, based on the same vaccinal, I'm sorry, this is not a word I've used before, vaccinological paradigm?
No, they are inactivated vaccines.
Oh, that's fascinating. Okay, which means that's live virus.
Right, but like whole virus that's inactivated and probably formulated in something that makes your immune system get like a kick. But they are not, they don't encompass the spike protein in the stabilized form, and they do not, obviously they're not mRNA technologies.
Gotcha, okay. Steve asks, do you think that that eventually we will have an annual shot like the flu shot?
I hope not. But if we do, I'm taking it.
Very good. All right.
You know, the flu is very different. You know, flu comes around every September, very different than what it came around in the previous year. So much so that your immune system, if you got the vaccine, just does not recognize it. Here, there's a lot more memory, and the hope is that with mass vaccination, we get to the point where the variants kind of subside, so much so that we aren't seeing drastic changes in the virus every single year, that it just completely evades the immune response. So far, we haven't seen the types of changes like in a flu virus, for example. And so it really is a combination of how the virus acts and the amount of immune waning. We are all on pins and needles waiting for, you know, looking at the immune responses as they wane in the people that have been boosted to kind of see. But hopefully the boost gave us a different set point and also decreased the slope of the waning a little bit.
Okay. My next question comes from Lamont, but I'm going to amalgamate this with a few other questions I've gotten. He asks, how are trial volunteers chosen? And then there's some other questions about like if both humans and animals. How do you choose the humans? How do you choose the animals?
So let's start with the animals, because that's kind of my thing. I mean, humans are cool, but so for animals, you choose your animal model based on the virus and does it replicate in that particular animal model. And not only does it replicate in that animal model, but does it replicate in the respiratory tract so that it is an example or akin to what we see in humans. And so that's how the particular mouse models that we chose very early on were chosen. And that is also how the non-human primates were chosen. And then, you know, the hamsters are they have very pathogenic models. So the virus kind of just takes off, replicates in the nose and also in the lung. But then also they lose weight, so you can get a very quick readout with the hamsters. And hamsters can transmit it from each other, so you can also assess transmission. So you basically, for animals, you look at the question that you want to ask, And then you pick and choose amongst the array of animal models that are available to you. And we're lucky to have so many here because there are some viruses that we don't even have really good animal models at all. Even the non-human primate isn't a good animal model. Like, you know, I don't know, I'm trying to think of one. Dengue, for example. And then for humans, clinical trial participants are chosen because they want to be there. They are given an informed consent about the study protocol and about their rights regarding their participation in the study. And they choose that this is the thing that they want to do with their body. And then as you get your pool of people who want to give in this way to the clinical trial, then you look at all of those people and you break up the demographics of the clinical trial so that it represents your population. So for example, the phase 3 clinical trial participants were chosen to mirror the United States's ethnic diversity and also the diversity of comorbidities like obesity and diabetes and et cetera.
If someone's interested in participating in these, are there like, do you have like an advertising budget? Do we contact you? How does that work?
I don't, because I don't do clinical trials, but I'm sure that the companies have a fairly hefty advertising budget. And then every single clinical trial that is registered under the United States government is on clinicaltrials.gov. It's actually like law that they be registered on clinicaltrials.gov. So you can go to clinicaltrials.gov. Say, for example, you have, you know, a friend who suffers from sickle cell and you want to participate as a healthy volunteer in sickle cell clinical trials. You can go to clinicaltrials.gov, search sickle cell healthy volunteer, wherever your city is, and call up a number and say, I want to participate in this clinical trial. So yeah, there is, I'm sure that advertising does a large bit and recruitment does a large bit, but there are people who really just wake up and say, I want to do this, and they go search for it.
All right, well, we have time for just a few more questions. I'm going to ask this question from Paul, and he asks, quote, can you explain why people of color are more susceptible to infection and adverse conditions to infection? Is solely due to a healthcare disparities and/or underlying conditions affecting many people of color? End quote.
As of today, that is what the data would suggest, that there is just health disparities and disproportionate amounts of comorbidities in people of color. And then obviously kind of sitting on top of that theory is just, you know, the social justice aspects, like the necessity to be going to work. And so you're more constantly exposed to the virus, or, you know, living in multi-generational households and et cetera, et cetera, et cetera.
Thank you. Okay. My next question is, I think, a very important question by our own Dr. Zolfilgrove here in biology. And she asks, she says, thank you for the presentation. She asks, what advice would you give students who feel overwhelmed by the long path to a career in research science?
You know, really build out your support network very early on. This career is certainly not something that it comes with so many rewards. I mean, but it is very long. Sometimes it can be very daunting. Science in general comes with a lot of no's before the yeses, right? You know, you get a paper rejected. I mean, the paper with the 2 proline mutations was rejected like 5 times before it was finally accepted, right? So, and you also have to as much as you can, just tell yourself that this is where you want to be, and recognize when it becomes too much and you realize that it just is not where you want to be. So listening to your heart, the only way you can really stay motivated is if it's easy to wake up and come to work every morning. I mean, I was like excited to like put this outfit on and do this today. So when that becomes a problem, then it might not necessarily be for you, but otherwise, being motivated requires that you certainly have a support system around you. And I would highly advise that 80% of that support system not be in science, because it's refreshing to be able to say to someone outside of science who, you know, knows you and knows your personality, to explain like, this is very hard, and them saying yes. And sometimes scientists are like, that's how it's supposed to be, like strap up your boots, and you know, and it's not always easy to hear that. And so and pay attention to one's own wellness is important. So I took a half day yesterday and I went and got this hair, and that's fine. Because I never take time off.
There are several compliments in the questions about your hair, by the way. So
you have to do that kind of for yourself and recognize when it's just far too much, right? And embedding whatever is your version of wellness into your daily life immediately is very important. And then the last thing that I like to remind people is that I literally wake up in the morning. This is something that my fiance kind of taught, because he does this with his nieces and nephews. I wake up in the morning, and when I go, the first time I see myself in the mirror, I say, you are smart, you are beautiful, and you have a voice. And so making sure that you affirm yourself into, I am a scientist every single day, really does help.
Yes, I have one more question for you, but that is just the most beautiful place to end. I just think it's so wonderful. What a great note to end on. Look in the mirror. I am smart. I am beautiful, and I have a voice.
And I have a voice.
Yes, what a wonderful message. I am so lucky to have been able to speak with you. I've read some of your readings and some of the things you've done, and I just feel so fortunate. I'm going to pass this back up to Rose. Thank you for your time with us today.
Thank you. Thank you.
Thank you, Dr. Corbett. You are smart, you are beautiful, and you have more than a voice. And thank you so very much. This has been wonderful. Clearly, you can tell from the questions, everyone was paying attention, and they had a lot to ask you. I would ask, we're about to close up, but I would ask if you would indulge us in staying on for 5 minutes after everybody else leaves to have an opportunity to chat with our president and maybe take a picture with her. That would be wonderful. Thank you so much.